All right. Hello. Welcome to the first symposium of the new year. I'm glad that you're here. We do have a few events and opportunities that are coming up that I wanted to announce. I just wanted to draw your attention to them. First, we are taking proposals for papers or posters for the National Collegiate Honors Conference. It's going to take place in New Orleans in November, so it's going to be a good time. In case you don't know, I take students to that, and we have fun. Like, we go and do stuff, and we eat out, as well as attending the conference. Keep in mind that if you have a paper or maybe a thesis in process, you can take that idea to this conference. That will earn you 100 points in academics, which is a badge at graduation, a line on your resume, and it's great professional experience workshopping your ideas. The link to submit will be found in Dr. Rookholm's email and on our Instagram and on our blog under Honors Forms. Also, we still have Anne Juliet tickets for next Wednesday. We just need you to bring $5, which I'm actually going to give back to you. Plant Hall 248. We have orchestra seats at the Stress. They're actually really close to the stage. So we'd love to have you come and hang out with us next Wednesday. It's coming up pretty soon. And then the Oxford Abroad applications are open and are due March 13th. So if you're thinking of that, mark your calendars to make sure that you get those applications in on time. And finally, the Honors Executive Council has a lot of exciting events this semester, from kayaking to serving with Metropolitan Ministries to volleyball, spin classes, silent book clubs. Our big end of the year event will be an Honors Prom, which is a semi-formal on April 10th at 6 p.m. in the gallery outside. We're going to have food, a DJ, decorations, whatever you can bring a friend or a date that is not in honors, as long as we get a head count. So we would love to have you come to our nerd prom. So now for today's guest. Dr. Fernandez-Figueroa is an assistant professor of environmental studies. As an aquatic ecologist, her research focuses on using remote sensing tools such as drones and satellites to spy on coastal systems. This monitoring data is used to train computer vision models, AI, to automatically identify coastal vegetation and track wildlife, such as sharks and rays. Her talk will focus on the application of these tools and how you can use them to satisfy your own scientific curiosity. Her talk is titled Spying on Nature, Drone Environmental Monitoring for the Curious. So please help me welcome Dr. Fernandez-Figueroa.
Hi, y'all. How you doing today? Good. I'm so glad that we're doing good. It's nice to see you so early in the semester. My name is Dr. Edna Fernandez-Figueroa. As mentioned, I am a professor of environmental studies here at the University of Tampa. I am in my third year, and I am having the best time. So I am going to share a little bit of my background about how I ended up using drones for spying on critters, because it was not an intentional road that I took. I think that I tried a bunch of different things until I found something that worked for me. Nobody taught me how to use these tools, but hopefully if this is something that interests you, today you will leave with a better idea about how you can incorporate these tools into your own life or research. So I am, by definition, an aquatic ecologist. What that means is that I am interested in aquatic systems at a very high level, okay? So I am interested not just on the animals that are in our ecosystems, I'm also interested in the plants, the chemistry, the physics, and how humans are affecting these systems. That is complicated. That takes taking into consideration a lot of different things. And it is aquatic because I have worked in both marine and freshwater environments. I got my bachelor's in marine science with focus in biology from Eckerd College, right over the bay. And then I went and pursued my PhD in fisheries, aquaculture, and aquatic sciences from Auburn University. In between those two degrees, I took a couple of years off and I worked doing coral restoration. I did that in the Florida Keys, in Puerto Rico, and in the Virgin Islands. And I mentioned that because if that is something that interests you, you should come talk to me so I can help you get a position and I can tell you about my experience because it was a really, really cool experience. Now, that being said, the process of getting from those degrees to where I am today was a little bit challenging. And we're going to start off this talk by doing a professional development exercise, pretty much because I want to get y'all a little bit more engaged. I know that we've been in class all day. So I am going to tell you a little bit about how I decided to choose the position I'm in today and the things that I prioritize. And then I want you to do the same reflection, and I brought some index cards for y'all. So when I graduated from my PhD, I needed to move back to St. Petersburg, Florida for personal reasons. And everybody told me that I was making a big mistake because it's very common for PhDs to kind of go to the place that they are hired. It's very hard for us to be location specific. I actually ended up working remotely for 2 years, which meant that I set up an entire water quality lab in my house. I had my drones, I had my microscopes, I had all my water chemistry equipment in my house. And then I would drive up to Alabama once a month to actually run a lot of my assays. And I realized that I did not really like that. And so this is essentially my reflection of what I wanted to choose for when I grew up. I wanted to make sure that whatever job I did had a positive impact. In particular, I really wanted to do applied research. What applied research means is that you are doing research in order to solve a problem. You're trying to be targeted. It's not just research because you're trying to learn more about that topic. It's not just science for the sake of science, which is incredibly important. But I really wanted to do something that I could build a tool and then somebody could take that tool and do something really cool with it and learn with it. Okay, so that's part of how I ended up here. The other thing that I was location specific, I had to be an hour away from my house or be remote. I really didn't want to be remote. And then I needed to have intellectual freedom. And that was really important to me because, as you'll learn today, I have a shiny object syndrome. I want to be able to go down whatever rabbit hole my heart wants to go down. And so I needed to work somewhere where I could ask whatever question I wanted and answer it myself if I wanted to. Now, those are my non-negotiables, and when I was looking for jobs, I didn't really care about anything else other than these 3 things. I could have been working in finance, and as long as I was meeting these 3 things, I would have been happy. And then I have my 3 other things that I really wanted to prioritize. The first one was traveling. I love traveling, especially for work. The picture that I have on the top left, I went and did research with some incredible scientists in Cuba, in Cienfuegos, and we went and we brought a bunch of equipment and we helped them bring new technology and new equipment to their labs, and we built it all in site, which was really fun. I've been all over the world, and it's all been on my employer's dime, and I really wanted to continue that in my professional life. I also wanted to have some time flexibility, primarily because the research that I do, I need to be able to just go whenever whenever the wind is good and whenever the sharks are out there, we're out there. And I wanted to have a positive work environment, and this was really important for me because I had been working kind of as a field grunt for a long time, and I wanted to make sure that it was a positive place where I could grow and do really cool things. The things I did to be able to do this, again, I was kind of chopping my legs by doing this because, again, there's a lot of PhDs. It's very challenging to be location specific. I intentionally created a research from my PhD, so my dissertation. You'll notice that none of my chapters talk to each other, and that's because I wanted to go to whatever employer was going to hire me and say, I can do that. So pretty much I've done everything under the sun, and when I interviewed for my jobs, anything they asked me, I was like, I can do that. And every time I would say I can do that, they would kind of stare at me like, this chick is crazy. But yes, I am definitely a jack of all trades and a master of none. That is a benefit and a detriment in some ways. I also was really good at diversifying my experience. I'm going to talk about this in a little bit. I did not really stay within the realm of academia. I tried to interact with the public as much as possible, and I had a bunch of fallbacks. Honestly, my plan was to start my own company and use my drones to help farmers grow food more efficiently if I didn't get a job. And that gave me a lot of ease of mind, because if I didn't find something that met these criteria, I know that I could do something for myself. So now what I want you to do is I have some index cards, and I want you to think about what your 3 non-negotiables are and what your 3 extras are. And then maybe some of y'all can share, and then maybe I can get to know you a little bit better before you get to know me, okay? Yes, please. Does anybody need a pencil, by the way? How are we feeling? Another minute? Or are we good? All right, so now you're going to find a friend and you're going to tell them what you wrote. Hopefully you'll meet somebody new. All right.
Wait, do I need to introduce it, or can I just like?
I would love to hear your name. That would be great.
Okay. My name is Emron Bar, but you can just call me Ron.
Okay, nice to meet you, Ron.
Nice to meet you too. For my non-negotiables, the first thing I said was exploration. I like my job to be involved with exploring the deep ocean.
Very cool.
My second one was a livable wage, because in America it would be a very hard life for me to live.
Mm-hmm.
And the third one was job security, because I'm scared of AI.
That's fair. Who's scared of AI? I'm scared of AI, and I make AI tools for other people to use. My AI is a different flavor of AI, I promise.
Microphone.
Oh, I'm sorry. Got yelled at. My type of AI is very different from the AI that you are probably used to. I do computer vision models. What you guys are probably used to is called generative AI. It is not the same thing at all. You are going to help me build a computer vision model at the end of this talk, okay? So we're going to do it together. You're going to see how it goes. Beautiful. Thank you so much for sharing. Would anybody else like to share? Yes, ma'am. Oh, thank you.
Hi, my name is Yulia. Some of my non-negotiables are intellectual stimulation, learning, like collaborative, open environment, and like feeling like I have an impact and like significance.
Amazing. Thank you very much for sharing. So this is kind of a strange exercise, but the reason I'm doing this right now is because when I graduated and I was looking for opportunities, I was being very specific, and I really should have been more broad. And if I would have had a list of non-negotiables, I would have probably found opportunities that were really fulfilling for me. But because I was so kind of narrow scoped when I first graduated with my bachelor's, I didn't have the ability to be like, well, actually, what really matters are these things. I didn't have money on this list. Money is important, and so are benefits. I'm not saying that that wasn't on my list. I needed to make money. I don't work for free, but not anymore at least. But these are really helpful, and I hope that you can use this reflection to help guide you as you do your job search in the future, okay? All right, so now that we have gotten to know each other a little bit better, I'm going to tell you a little bit something about finding opportunities. I have done all kinds of crazy things, and I'm going to keep this limited because I know that we have people who are not scientists in the room. But I would say do not limit yourself to jobs that you think you're not qualified for. If it fits your criteria, go for it. Let people know you're looking. Put it in your conference slides. If you go to a conference, post it on Instagram. I love making YouTube videos explaining to people how to work with drone imagery. I have gotten some really cool contacts doing that way. Networking is hard when people in your field aren't close to you. Doing it virtually can be really, really powerful. And then there's these weird thing called informational interviews. If there's somebody who has a job that you really want, you can ask them for a 20-minute interview where you just ask them what it is that they do. Then they'll know that you're interested, and maybe the next time a job gets posted, they're gonna call you first. If you are an environmental studies, marine science, any kind of ecology-related person, and you're looking for jobs, please check out the Texas A&M Natural Resource Job Board. There are crazy opportunities in there, okay? So that was just my disclaimer for y'all. The other thing I want to tell you is find some mentors that you trust. These are just like some of my mentors. These are people who I asked all of them. I asked them the same question. Every single one of them gives me a different answer based on their expertise. Then I get to make up my own mind. I don't have to do what my mentors tell me, but have people that you trust that can help guide you through your process because it's really challenging. And the last thing is share your interests often and do weird things that actually are representative of your passions because that's where you actually have the most impact. So one of my favorite things I've ever done is I used to go into prisons and talk to inmates about water quality. That was super cool. I am definitely afraid of public speaking. That opportunity made it so I am way more confident in public speaking now. There's like some other random things in here. I really love to do outreach, primarily with kids, because that's usually who we're doing volunteering with. But I also used to bring volunteer opportunities to retirement homes so that retirement people could learn how to monitor water quality. And I also bring a lot of science to music festivals because that's a way in which you can capture people who are kind of in that 20 to 30-year range for outreach. And I mention this because the more of these things that you go to, the more exposure you get, the more networking that you do. Oh, and then the last one is exposure therapy. Again, I hate public speaking, and yet here I am. I'm a professor. I public speak every single day. I worked through all of the nausea and the heavy breathing and all the things that needed to happen so that I could present. Do not let the things that make you nervous and anxious keep you from following your dreams. Like just if you are in a safe place, go for it. Do what you need to do and get those cool experiences. All right, so now we're gonna talk about research. So I'm going to tell you a little bit about my dissertation research, primarily because I want to tell you why I didn't end up doing any of the things that my advisor wanted me to do. I did complete them and I did publish them, but every time I would finish a project, I would say, this isn't for me, but now I learned all those skills and I know I can market myself for them if I need to. The first project that I did was working with these little teeny tiny zooplankton called Daphnia. They are commonly known as water fleas. They are super cool because they are clonal, which means that when everything is peachy in the water, the females, it's all girls in the water, and they just make clones of themselves. They are genetic clones of their mamas, okay? When things start to get a little bit dicey, usually when it becomes winter or when it gets too hot for them to be able to survive or there's something toxic in the water, the females will make a male and they will mate with them and they will create what's known as an ephypia or a resting egg. That egg can stay underground in the sediment for hundreds of years. And what's really cool about it is that that gives you a genetic representation of what Daphnia looked like 100 years ago. So what I did for my dissertation was I went in and I found Daphnia eggs that were around hundreds of years ago when we didn't have climate change and we didn't have a bunch of harmful algal blooms, a bunch of toxic algae in the water. And I took those eggs and I hatched them. And then I also hatched some Daphnia from today. And those are my super Daphnia that are always exposed to toxins, they're always exposed to harmful algal blooms, and it's hot climate change. And I wanted to see if the Daphnia from the past behaved different from my super Daphnia today. We knew already from the research that my advisor had done that the super Daphnia did really well when we fed them toxic cyanobacteria because they were adapted to it. They had been exposed to it for a very long time. However, we didn't know what would happen if we added additional stressors, and that was the point of my research. On this slide over here, I talk about trade-offs. When you think about a trade-off, I want you to think about an adaptation that could be beneficial in some times, but it can also be detrimental in others. So being really tall might be really beneficial if you need to reach something at the top of the shelf, but it's not gonna be very great if you're in an airplane and you're trying to fit in one of those tiny seats. There's always going to be a trade-off for every adaptation, okay? So my hypothesis was that being really good at tolerating toxic cyanobacteria would make it so that you are not as competitive or fit when climate change happens and things get warmer. Okay, so I went in and I exposed my Daphnia to a bunch of different temperature treatments and a bunch of different toxic algae. I had over 85 treatments. I was in the lab a lot. And what I found was that there was no energetic trade-off. A negative result is still a result. But what I learned from this experience is that I do not like working in the lab. I am an environmental studies professor. There was a lot of waste that happened through this process, and I just couldn't really identify with it. There's absolutely nothing wrong with doing lab research. It is super important for informing things in the field. But personally, I'm too type B to do something like this. But I did learn a lot about things like culturing algae, culturing the Daphnia, getting those eggs from the sediment and resuscitating them was really cool. As well as some genetics. So I got to learn a bunch of different things, more than anything that I don't like to be in the lab. So then what I did was I went and I sampled every single drinking water utility in Alabama for 2 years. So these are all of our surface drinking water utilities. What do you guys see missing in this picture? Where are these people getting their water? From the bottom. Surface water means we're getting our water from a lake or a river. What? Yeah, we're getting our water from aquifers in the lower portion of Alabama, so I didn't have to worry about driving all the way down there. But I had to work really closely with our utility workers, and they would go and they would collect their samples for me, and I would run them in the lab. Sometimes they only send me one a year. Sometimes they sent me one a week, and I had to process all those samples. And I loved it. I found that our water was actually pretty clean. We had a lot of nutrients in the water, which should mean toxic cyanobacteria, but we didn't have any toxic cyanobacteria, which was great. So I got to give our utility operator some positive news, which is always awesome. But we did find that there was a potential for there to be toxic cyanobacteria because there was so much nutrients in the water. We do not want toxic cyanobacteria in the water because it makes it way more expensive for us to treat our drinking water before it gets to us. It can also close down the water utility, meaning that people in that area do not have access to drinking water. And cyanobacteria also produce these things called taste and odor compounds that are not toxic, but they make the water taste like mud, and it's really hard to remove it from the water. So those poor utility officers, when they had algal blooms, maybe the water was safe to drink, but it smelled and tasted like mud, so they were just getting calls constantly. So I was trying to find data to help inform why this was happening so we could control it. Nothing happened when I was sampling, which is how research works. But it was awesome. I learned that I like to collect my own data. It was when you're processing samples somebody else sent you, you don't know if something happened when they were handling that sample, when they were sending it to you, what happened in that process. If you collected that own sample, you can say, yeah, that was a sample where I didn't take that Secchi reading too well, so maybe I shouldn't trust it. You don't really have the ability to do that when you're working in this giant team of hundreds of people. But it was really cool, and I got to do GIS, which is my favorite. It's a mapping software, which is why I have this lovely map. Then COVID happened, and I was supposed to go to Canada and do a bunch of field work. I obviously could not, so I needed to graduate, so I went in and I collated, so I downloaded data for over 1,000 lakes, and I looked at how their algae had changed through time, because I was doing my PhD, so I was reading hundreds of papers all the time, and every paper had something different to say about harmful algal blooms. They're getting better, they're getting worse, they're the same. And I was like, well, what's the reality of the situation? And I realized that the reason why everyone had a different story was because everyone was saying how things had changed without looking at what things looked like at the beginning, right? Change is relevant from the start point. So what I found was that in the past 30 years since I started my collection, most of the lakes were green, and they have stayed green. So out of the 1,000 lakes, 546 of them were green and had toxic algae in them. But what was super interesting was the ones that were super duper green and toxic were actually getting a lot better. But the ones that were clear and nice and pristine and that you could go and play in and get your drinking water from, those are getting a lot greener. And those are the ones that we are really worried about. So then I did some really cool analyses, and I was able to show areas in which the water quality was getting a lot better and a lot worse. And then we can now use this to inform policy, so people in those areas can say, hey, we need more money for managing harmful algal blooms. This is an emergent issue. And this is where I really realized I really want to do applied research. I want to do things that people can take my data and show it to the government and be like, give me money. So this is kind of where that drive and that excitement started for applied research. And then I discovered my drones. So at that time, I was working really closely with catfish farmers because Alabama is one of the biggest producers of catfish in the US, actually in the world. And so I was going out there and I was collecting water quality data for my farmers, but a lot of their farms have over 200 ponds, and these are experimental ponds. Their ponds are big, so they're spending 2-3 hours going out and just collecting things like oxygen. Going out and collecting things like toxic cyanobacteria, that is something they can't even run that analysis by themselves. They have to collect the sample and send it to us. So what the farmers were doing was whenever a pond was green, they were just nuking it with toxins to kill all the algae. That's not great. We don't want catfish that is just doused in copper. So I was like, okay, what if I built you a drone that you could fly over your farm, and the drone tells you how much algae is in the water, and then based on the drone reading, you can decide whether or not you want to treat it with chemicals, okay? So that's what I did. So I had no idea how to fly a drone. My advisor didn't know how to fly a drone. I didn't know nothing, zero things about drone. I went camping with my now husband, and I knew about satellites. Satellites are used very often to estimate the health of plants. And we were out camping, and the leaves were changing color, and I was like, I wonder if I could use a drone to see how the leaves are changing color and the relative abundance of pigments. And I went on my phone, and I was like, harmful algal bloom drone. A review paper had been published that day. And I read the whole paper, and I was like, this is for me. We're doing this. So I went out. I had some funding from the National Science Foundation, and I bought 2 drones. They were little phantom drones, and I attached 2 sensors to them. I had these sensors here because I wasn't just trying to take pictures of the water, I was trying to estimate how much algae was in there, which is not simple. So the graph that you see on the map, those are my 3 sensors that I tested. The drone on the left, it's like a big white drone. I had 2 of those, and then I had one that had the green camera in the middle, and the other one had a red camera attached to it, okay? The one on the far left is a regular, what we call a red, green, blue sensor. It's basically the sensor that you have on your camera. And the lines that you see are the reflectance and absorptions of the different pigments that we find in algae. Don't worry about that too much. The idea is that chlorophyll reflects some colors and absorbs others, and we can take advantage of that difference to see how healthy the plants are or how much algae is in the water, okay? So the colors that you see, can I do this? Haha. These little bands that you see sticking up, which are these little colors, those are the colors that the drone is able to measure, okay? So this one's able to measure red, green, blue. This one can look at green, blue, and then near infrared, which is really important because near infrared plants can't absorb it. And then I had another one that was really expensive that could measure green, red edge, which is another pigment or wavelength, and then near infrared. This red one was like 10 grand. It was really expensive, and I was really hoping that it wasn't gonna be the best, but it was. So out of all the drones and all the sensors that I tried, which was 4 of them, the one that can measure the near infrared was the best performing one. For reference, this one was 10 grand, this camera was $300. So it was a really big difference in price, and I was really hoping this one would work, but it did not. And essentially what I'm showing you here is this is how you calculate the amount of vegetation in the water, and these are essentially the calculations that I used, and you can see that the drone did really, really well at seeing how much algae is in the water, but it didn't do as well at estimating how much toxic cyanobacteria is in the water. Those are 2 different things. Regular algae is fine. It's not gonna hurt the fish with some caveats. Toxic cyanobacteria is what I was really, really interested in, and I found that they could use it to a certain extent. And this was really cool and kind of catapulted my love of using drones for answering questions. At this point, this is my last chapter of my PhD. Everyone was mad at me because none of my chapters talked to each other. But whatever, I graduated and I got my job in doing my PhD here in Tampa, or my postdoc. When I moved here, I was supposed to have a month off between my PhD and my postdoc, and we had this horrible fish kill in Tampa Bay. So in 2021, we had over 3 tons of dead fish wash up on shore. It was horrible, and I was like, okay, well, I'm here, why not collect some data? the first thing that I did was I obviously did a lit review. I wanted to know what we knew about fish kills here in Florida, and I found out that we didn't really know very much. So I went in and I did a review of all of the fish kill data that we have for the state of Florida. And what I found is that there's this thing called the Fish Kill Hotline where you call in and you say, hey, I have a bunch of dead fish here. Come check it out, FWC. What I found is that the Fish Kill Hotline is really cool if you're trying to figure out where fish kills are happening, but they don't really tell you very much about what kind of critters are there or how many of them. We did find that people have been using the hotline more in current, like as time has gone on, and that most people were just submitting their reports from docks. Nobody was really looking at what's going on offshore. And then the best thing about this project was that we found out people were only really reporting like fancy species, charismatic megafauna. So anything that was a fishery species, anything that was a manatee, a turtle, those things are getting reported like crazy, but nobody really cared about the tiny things. And if you look at this picture right here, the majority of the things are tiny things. So we were really missing a really big part of that story, and that was really cool to be able to document. How did I figure out how many of the tiny fish were in there? A drone. So as I was doing this, I was going out and I was flying my drone and collecting samples, and then I had to actually process all this data. I'm gonna give you 30 seconds, and I want you to try as hard as you can to count the fish that are in this image. Wait, there you go. All right, what do we think? Was that easy? No. Did you think I was having a good time trying to count all these fish? No, I was not having a good time, so I had to figure out a better way to do it, and that's how the AI came to be. So I needed to figure out a way to have the computer help me count all these gosh darn dead fish before I lost my eyesight. There are ways to estimate, so you could theoretically go in and put down some targets, which I did, and extrapolate the number of dead fish in that target to the rest of the beach. But as you can see, the distribution of these fish is not very regular. So if you put the target in the wrong spot, you would severely underestimate the amount of dead fish in that area. So what I did was, this is what my flights look like when I put them together. The way the drone flies, I'm not manually flying it. I have a mapping software that basically I tell it, hey, I want you to fly at this altitude. I want this much overlap between my images. And then I have to go in and do something called photogrammetry and actually take all those thousands of pictures and turn them into one big picture, okay? So I went in, I did all that processing. You can see there was an insane amount of dead fish on some of the days. And then I built a computer vision model to then automatically identify and enumerate my fish. And then I didn't completely lose my eyesight, which was great. Now, the AI obviously did not know what a dead fish looks like. It's not trained for that. Primarily, we train computer vision models to look at things like cars and stoplights and people, and to recognize your face when you're at Walmart to see if you're gonna buy that shampoo or not. And yes, there's literally, in the program that I use, there's literally a button that I can press if I want to look at people's like faces and see how they're feeling, which is really strange. But there was no library for what a dead fish looks like, so the majority of this research was me going in and tagging a bunch of images and teaching the computer what a dead fish looks like. That sounds kind of strange, but the machine only learns what you teach it, okay? So if every single image that you captured was of a dead fish over sand, it assumes that dead fish are only found over sand, and they would never look at a dead fish that's over a rock. And that's why I have these three different systems, right? I have one that's really sandy, one that has a bunch of seagrass, and one that has the rocks. That's because that makes the background different and makes it so that the drone or the computer learns what a dead fish looks like in a bunch of different settings, okay? All right, cool. So this is what I did for my postdoc, and now we're gonna talk about what I do here in Tampa. So, La cucaracha, because she kind of looks like a little cockroach. This is a really cool drone because it has the ability to measure not just a normal, did I open her up? Yeah. All right, so this is a really cool drone. This drone is originally designed for military purposes. For military purposes, and that's the only kind of drone that I can actually buy with federal funding. The drone has 3 special cameras. One is a super zoom, so I can really, really creep on critters. One is a regular wide lens, and then I have a thermal camera, okay, so I can look at all kinds of things. We have 2 other drones. I'm gonna show it to you while I put this back. And there's no audio, so it's okay. This is my other drone. It's called a vertical takeoff and landing drone. What it does is it goes up vertically, and then once it reaches its target altitude, it's going to level up and then go. It flies all on its own. I am actually not allowed to touch, well, I'm allowed, but there's nothing I can do once that drone is in the air. I tell it what to do. It does it all on its own and then comes back to me at the end. It's very scary. This drone I actually manipulate myself. It's fine. And what I am currently trying to do with my students is we are trying to map coastal vegetation in Tampa Bay. So I want to be able to fly my drone over any coastal system and then feed that picture into the model and have it tell me there's these many white mangroves, these many black mangroves, there's these many invasive species present, et cetera, et cetera. The way that we do this is we go out, we collect our thousands of images. You can see this is what one of our flight plans looks like. So we go, we make a grid, and we fly over the same spot a couple of different times from a bunch of different angles. We go in, we take all those images, we make a giant picture with it, and then we go in by hand and we tell it, hey, this is what a cabbage palm looks like, this is what a palm tree looks like, this is what just water looks like. And we go in and we segment all of our images. And then we tell the computer, this is what vegetation looks like. And then we get some feedback, and we are able to then calculate how much of each vegetation is covering that area, okay? I'm doing this research with Delaney. She's one of my students in the Environmental Studies department, and it's really fun if you want to learn more about this. We are hoping that because we're collecting this data with our thermal drone, I know for a fact that there's a bobcat that lives in this area, and we're hoping that the thermal drone will help us figure out where it is in the morning because it has a thermal temperature, like a signature. So we're hoping to combine our thermal drone and some hunting cameras so that we can track this population of bobcats. The other thing that I'm working on, which I'm working on with my GIS students actually, is we are going through and we are automatically estimating the amount of seagrass scarring from satellite imagery. So we have satellites that are always going over our planet. They're constantly collecting information, and we would like to use that to see how seagrass scarring, how it has changed through time, but also how long it takes our seagrass to recover. For this research, we are using our regular drone, but also our underwater drone. This is essentially a teeny tiny submarine or an ROV. And our plan is to fly the drone overhead and then fly this guy underwater, and hopefully, so in combination of our satellite imagery, be able to actually identify what species of seagrass is in those different areas. All right? All right, and then the research that my students are working on, I gave them free rein on this one, is we are trying to utilize a drone to automatically identify, so what species of shark is it, or stingray. So not only identify what it is, but also see how it moves through the water. So things like tail beat frequency, how many times is the shark moving its tail as it moves through the water, and also how fast is it moving. We're hoping that that data is gonna give us a little bit of understanding about their metabolism, for example. Our hope is to go to the east coast of Florida and monitor the blacktip shark migration. There's hundreds, if not thousands, of blacktips that move down the coast of eastern Florida in the spring. And our hope is to take that drone from up ahead and look at them, but also take the underwater drone and look at them from below, and hopefully be able to identify whether it's a male or a female. We'll see. We've never tried it before. This is all new stuff. It's really cool to do this kind of stuff. You gotta code a lot. Nobody taught me how to code. I figured it out as I was going. It was not a pleasant experience, so if I can give you any kind of advice, if you can learn how to code, take advantage of that opportunity. But the idea is we then tag the different parts of the shark as it moves through the water, like you can see down here, and then we can make these really cool graphs that show us how it's moving. We are also doing this with our resident population of cow nose rays. Well, they're not resident, they move around. And these are some of my students that have been helping me with that research. Because it takes quite a bit of time and the visibility isn't great in Tampa Bay, a lot of the research gets kind of passed on from one student to the other. We're hoping that going to the East Coast will help with that because the visibility is much better. All right, how are we feeling? Specific questions or concerns right now that we want to share? Should we tag some images? Yeah. Okay, so if you scan this QR code on the top left, it is going to take you to a website that looks like this, very scary. You're going to click right here because it's fine. You can get in there. And you're going to just put in your email and whatever password you want to make, it doesn't matter. And then you're going to teach the drone what stingrays, sharks, or fish look like. There are some dummy ones in there. There's some dolphins. There's some manatees. Don't tag those ones. Those aren't sharks, stingrays, or fish. And the way that you tag on this software is you're going to, with your finger, you're going to click what it is, and then you're going to click in one corner, nothing's going to happen, but something has happened, and then you're going to click the other corner of that box, and then you have now tagged that image. Now when we go to build our model, you will have helped us build our training library, and if you would like, we will definitely acknowledge you in the paper, and if you want to learn more, you come talk to me and we can get you involved, okay? So I'm going to give you all maybe like 2 or 3 minutes to do this. Are we running out of time? Okay, so I'm going to give you a minute, and then 2 minutes for questions. Or you can ask questions as we're doing this. Does anybody have any questions? Wow, I was thorough. All right. And while y'all are doing that, I'm going to give you all an announcement. If you want to come do outreach with me, I'm going to do a similar activity to this part and then some cool arts and crafts at the St. Pete Science Festival in St. Pete on Saturday. So if you want to come interact with some people and do a little volunteering, I'm going to be there from 9 to 4, but you don't have to be there all day. You can come hang out with me for an hour or two and then go play in St. Pete. But it's a really cool event. If you have never been, it's really cool. Did it work for everyone? Is somebody not working? Did the link not work? Can you see it? Jessica, tell me the truth. No! Actually cooler. That's okay, we're learning. This is the first time I've tried to do this with a group, so thank you for being my guinea pigs. We are officially out of time, but it has been such a pleasure getting to know y'all. If you ever need anything, my office is in Grand Center 112, and I would love to get to know you. All right, bye y'all.